Hello, everyone. Today we have a topic that most of us find very, very interesting. We're going to be looking at sleep. But we're going to look at sleep within the context of a structure in the brain known as the reticular formation. So today's session is entitled The Reticular Formation, Sleep, and Coma. So what is the reticular formation? Well, as you can see on the diagram, the reticular formation is found within the brain stem, and it represents an arrangement of neurons that seem to be connected, running from the top of the spinal cord through the brain stem towards the midbrain. It doesn't seem to be a singular structure with well-defined margins and boundaries. Rather, as I mentioned, it's a collection of neurons that are arranged in neural nets. Because of that, it's been very difficult to define historically. And only as we've developed new histological techniques have we been able to put more definition and more structure to what parts or what components make up the reticular formation. What we do know, particularly about the reticular formation, is it has many, many functions, key to which is arousal and regulation of sleep. So based on our knowledge today, we can say that the reticular formation is composed of three different types of neurons. There are short interneurons within the brainstem that form circuits, very similar to the central pattern generators in the spinal cord involved in locomotion. These generate motor signals and motor rhythms for stereotype motor activity like facial expressions, eating, swallowing, and breathing. Then there are interneurons in the thalamus and networks that are very important for sensory perception and arousal. Here we see how sensory perception is linked to arousal. For example, a baby's cry in the night wakening a parent. And finally, this part of the reticular formation that we might best understand are long projection neurons, where the origination, the nuclei, the collection of cell bodies are located within the reticular formation. And then they send projections out throughout the entire brain, and in some instances down into the spinal cord. These projection neurons are found to be in groups of clusters based upon the neurotransmitter they release. And we now know we have projection neurons for serotonin, for noradrenaline and adrenaline, for acetylcholine, and for histamine. When you look at the arrangement of these nuclei, we find that it is divided into three columns from an anatomical point of view. The median column, that which is in the midline, is the raphe nuclei that are associated with serotonergic signaling. The medial column is called the magnocellular nuclei, and the lateral column is called the parvocellular nuclei. And these each have different roles and different functions, which you will study in more detail in your anatomy courses. So let's talk a little bit about some of the functions of the reticular formation. And if you've been following my channel and listening to other lectures, we would have discussed many of these before in different contexts. So one of the key roles of the reticular formation is it helps to regulate somatic motor movements. And there are two particular tracks here, one that originates in the pontine reticular formation and one that originates in the medullary reticular formation. And so this gives rise to the pontine reticulospinal tract and the medullary reticulospinal tract. Now, these two tracts seem to work in conjunction with each other, with the pontine tract being primarily excitatory and the medullary tract being primarily inhibitory. And the balance of these two tracts seems to be particularly important in helping to regulate posture and to help regulate balance. These tracts also seem to have an important role in regulating the central pattern generators that I spoke to you about a little bit earlier, that are involved in things like breathing and swallowing. And they do seem to be connected into the centers that control eyeball movements. Then you would have looked at in other parts of your studies, the fact that lying within the reticular formation are many centers that are important for regulation of the cardiovascular and the respiratory system. We don't have time to look at these things now, but of course, if there's damage to the reticular formation in the brainstem, these areas could be compromised, and that can have catastrophic consequences. Again, something we would have seen already is the fact that certain of the long projection neurons from the reticular formation are able to descend through the spinal cord and then affect the perception of pain. This is called the descending analgesic system, and it's an extremely powerful analgesic system, able to remove pain to a large degree. For example, soldiers on the battlefield. The key neurotransmitters believed to be involved here are the serotonergic system and the raphe nuclei, and the noradrenergic system and the locus coeruleus. And then finally, the function that the reticular formation is best known for, why we even talk about something called the reticular activating system, is attention, arousal, sleep, and consciousness. These projection neurons that we spoke about, they seem to be involved in sending signals up to the cortex and ensuring that we stay alert and also focus on particular things. So for example, I mentioned to you previously the fact that a parent can be sleeping in the night but is awakened by the baby's cry. That seems to be the ability of sensory signals to activate this arousal system and focus attention upon those signals. And so you can see there the simplified flow of information that's involved in attention and arousal, where sensory input will activate these long projection neurons. And then via the thalamus, this will cause arousal of the cortex. And of course, interruption of these pathways could then lead to unresponsive states like a coma. Similarly, these projection neurons then are involved in sleep and what we perceive as consciousness. We know a lot more about the neurobiology of sleep, and scientists are also probing and seeking to understand the neurobiology of consciousness and how these projection neurons can be involved in that strange thing in the human experience that we call consciousness. So having listed the functions, let's talk a little bit about some of the situations in which this is important. And we want to start off with a clinical situation called a coma. There's a funny image there of the cat. It says, oh, another PowerPoint. Hang on while I slip into something more comfortable, like a coma. So what is a coma? Well, it's an unarousable, unresponsive state in which the patient lies with the eyes closed. In this state, the EEG is usually abnormal, indicating that functioning of the brain is not quite as it should be. But many of the reflexes associated with the brain and central functions may still be present. For example, withdrawal reflexes and the vestibulo-ocular reflexes, indicating that these are controlled at a lower level within the central nervous system. What would cause a coma? Well, we now know that a coma can be caused by either damage to the ascending reticular activating system. And this can happen due to compression, some situation that is compressing the reticular formation within the brainstem, like a tumor or cerebral edema. Or it can be the result of a stroke, interruption of the blood supply to those particular regions. These are the most common causes for coma and usually might occur following traumatic injury or, like I said, long-standing tumors. Less common would be damage to the cerebral hemispheres. But for this to produce a coma, you'd have to have a very extensive lesion that's affecting large areas of the cerebral hemispheres. And this often does not occur all at once. So you can see that on this diagram here. Just by looking at the anatomy, it's much easier due to the location and the small area that the reticular formation occupies for damage in this region to cause a coma than for large sections of the cortex to be lesioned or knocked out. When this does happen, patients often enter into what are called impaired or vegetative states, an area or an understanding of consciousness that we don't quite fully comprehend at this point in time. Now, to help us understand comas from a clinical point of view and how to best assess and manage them, clinicians more than 40 years ago in Glasgow, Scotland, developed a scale, the Glasgow Coma Scale, to look at the state of consciousness of a person. So it's a neurological scale and it's designed to provide both a reliable and an objective way of recording the state of consciousness. And what it's supposed to do is standardize how we treat and manage persons in a coma. You can see by the image on the right of your screen that three things are assessed: eye-opening response, verbal response, and motor response. And each of these is given a score. Note that the eye-opening is from 1 to 4, the verbal is from 1 to 5, and the motor is from 1 to 6. And note in all cases, absence of the response totally receives a score of 1 and not 0. And that means the lowest score that you can receive on the Glasgow Coma Scale is 1. The eye-opening responses are the person will not open their eyes whatsoever, as nothing can cause them to open their eyes. And it ranges all the way to they spontaneously open their eyes on their own, which will receive a score of 4. The verbal response also is there's absolutely no verbal responses. Moving towards incomprehensible sounds, words that aren't being put together in sentences. Person is speaking in sentences, but they're confused and they're not sure where they are. And finally, arriving at a place where they're oriented and they're very clear and they're able to communicate. And then the motor responses, there's absolutely no motor response. Then they have extension responses, abnormal and normal flexion responses, and finally they can obey commands. This allows us to then come up with 3 grades rating the severity of a coma, and you can see the scoring pattern there. A mild coma would be a score of 13 to 15, a moderate 9 to 12, and a severe anywhere from 3 to 8. And like I said, when you get into your clinical part of your training, for those of you who are studying medicine, this will have a lot more relevance for you. But it's important to appreciate it now. Now, within this context of looking at the coma, I want to talk about 2 abnormal postures that can be adopted during damage to reticular formation and during a coma, because they help us understand some key physiology. The first is what we call decorticate rigidity, and the second is called deliberate posturing. And you can see the differences right there. You can see in the decorticate, the arms are adducted and flexed, and the legs, the lower limbs, are internally rotated, and there's plantar flexion of the foot. In contrast, in decerebrate posturing, the arms are extended and pronated, while the legs are also internally rotated and flexed. What's the reason for these differences, and what does it tell us about what's taking place? Well, if you have decorticate posturing, that means there's damage above the red nucleus. And if you have decerebrate posturing, that means there's damage below the red nucleus. Why is the red nucleus important in these different responses? Well, let's remind ourselves of the rubrospinal tract, which originates in the red nucleus and then synapses primarily on the alpha motor neurons in the cervical and the thoracic spinal cord. Now, this rubrospinal tract is basically excitatory to the flexor muscles of the upper limbs, and it inhibits the extensor muscles. Okay? So it excites the flexors and it inhibits the extensors. And this tract is usually inhibited, it's usually regulated by the higher cortical centers, and therefore you don't see it in action. So the higher cortical centers inhibit the rubrospinal tract. Now, if you have a lesion above the rubrospinal tract, you have removed the input of the higher cortical centers. And so the rubrospinal tract, it now takes over. And what does it do? Remember, we said it produces flexion of the upper limbs, hence decorticate rigidity. If you have a lesion lower down in the brainstem, below the rubrospinal tract, well, what are we now going to see? What are we now going to see? I want you to pause and think about that. Try and work it out. What you think is going to happen due to the lesion below the rubrospinal tract? I'm going to play this video while you think about that.
Patient is in the decorticate position with the upper extremities in flexion, lower extremities in extension, reflecting that the brainstem motor centers are working, but we don't have modulation of those centers from the corticospinal tracts and their connections to the brainstem centers. If we now go a step lower as far as the level of dysfunction and eliminate the function of the rubrospinals, we go from decorticate to decerebrate posturing. The lower extremities stay in extension. Now the upper extremities are in extension because we don't have any type of regulation or modulation of the vestibulospinal or the reticulospinal tracts, but we've eliminated the influence of the rubrospinal tracts at this point in time. If there's to be further progression and the patient has further deterioration, and we now eliminate the function of the vestibulospinal and the reticulospinal tracts, the patient would then become flaccid.
So hopefully you heard the answer there if you hadn't worked it out. If you have a lesion below the level of the red nucleus, what we have now done is we've removed both the upper centers and the activity of the rubrospinal tract. And therefore the activity of the vestibulospinal tracts begin to take over. And that leads to the decerebrate posturing and the extension that we've seen there. And as you heard in the video, anything lower than that and worse than that essentially can lead to death. And so these postures, these typical postures, given us an idea of where a lesion might be within the brainstem and the reticular formation, and therefore also give us an idea of the severity, with the decerebrate being a little bit more severe than the decorticate, if you look at the motor response on the Glasgow Coma scale. Okay, so we've spoken a little bit about coma, so let's talk a little bit about sleep. So sleep is a characteristic immobile posture in which you have diminished but readily reversible sensitivity to external stimuli. Now, that definition might not be very useful, and defining sleep can be quite difficult. But what we do know is sleep is very important. If human beings don't get enough sleep, it affects their performance, and it can cause severe mental illness. And in animal studies, a lack of sleep or sleep deprivation can even lead to death. So we know sleep is very important. We know it's an essential part of almost all of life, not just human life. But we're still trying to understand what is the function of the sleep. And here we have some ideas of what's going on during sleep. It has been suggested that it's a time when there's a clearing of the waste products, that are metabolic products due to activity throughout the day can accumulate in the brain and they need to be cleared. It has been suggested that there's a restoration of immune function linked to clearing of those products and resetting particular circuits in the brain. We know that sleep is important for memory consolidation, and it's also important for good growth and development, especially in early life. And we know it seems to be important for regulating the ANS and mood regulation. So based on the fact that when we have a lack of sleep, all of these things go wrong, it has been suggested that these are important functions of sleep. And I think that's a reasonable projection because, as I said, when you lack sleep, all of these things begin to go wrong. Now, this diagram just shows us on the left that different animals sleep different amounts during the day. That's just interesting to see that a bat can spend up to 20 hours of a day sleeping, whereas domestic animals like cattle, they sleep less than 4 hours a day. Many studies have now shown that for the human, the average human needs between 7 and 8 hours sleep a day. And if you get too much sleep, or if you get too little sleep, based on epidemiological studies, there are consequences for your health. The other thing we note is there's a physiological reduction in total sleep time with age. A newborn might spend about 16 hours a day sleeping, but by the time you reach 70 or 80, that may have reduced to about 6 hours. And many of you might know your grandparents who might be early risers or might have complained of difficulty sleeping. There are certain physiological changes that we're trying to understand, in which the sleep cycle changes, and it's much more difficult for older persons to both fall asleep and stay asleep. You will notice in that diagram on the right that the big change between the newborn and the adult is the amount of time they spend in something called REM sleep. I will explain what that is in a little while. And just an interesting fact about sleep, because sleep is something that fascinates all of us. Here's a dolphin, and dolphins sleep one hemisphere at a time. Because as you know, dolphins are marine animals, but they're mammals, and they have to come up to breathe. And therefore, to ensure that they're able to do that, both hemispheres can never go to sleep. And so you can see there's this interchange where both hemispheres can be awake at the same time, but you never have a situation where both hemispheres are asleep. One hemisphere is always awake, controlling the movement and the regulation of the dolphin. How do we track sleep? Well, this is done utilizing a series of markers, primarily the EEG, also the EOG, a measurement of eye movements, and measuring your ANS, your respiratory rate, your heart rate, your skin conductors. Based upon that, there's a typical sleep cycle. Now, the typical sleep cycle consists of slow wave sleep or non-REM sleep and REM sleep. REM stands for rapid eye movement. Older classifications divided slow wave sleep into 4 stages, but the American Society of Sleep Medicine recently put out a new classification, which is widely being adopted, in which slow wave sleep is now classified into 3 stages, and that's what I'm utilizing here. And you can see how the sleep cycle moves throughout the night. You move from the awake state into what's called non-REM stage 1, and you progressively go into deeper sleep, into non-REM 2 and non-REM 3. Non-REM 3 is usually called deep sleep. Now, after non-REM 3, you then move back to non-REM 2, non-REM 1, and it's only then that you move into REM sleep. And that would then complete one sleep cycle, which takes between 90 and 120 minutes. So the cycle is non-REM 1, 2, 3, back up through 2 and 1, and then you have REM sleep. Not non-REM 1, 2, 3, and then REM sleep. And this cycle repeats itself throughout the night, 5 or 6 times. But what you'll notice as you approach morning, sometimes you don't go into deep sleep, and the cycles get a little bit shorter. Now, these cycles have been described based upon diagnostic or particular characteristics of the EEG. When you're awake, we tend to have low amplitude, high frequency waves known as beta waves, anywhere from 14 to 30 hertz. If you become drowsy and you close your eyes, especially over the occipital lobe, you see a slowing down of the EEG into what we call alpha waves, 8 to 13 hertz. And these appear during stage 1 sleep, as well as the slower theta waves. As you go deeper into sleep and you reach stage 2, you begin to see unique features of the sleep EEG, known as sleep spindles and K complexes that you can see on the diagram. And finally, when you move into non-REM stage sleep 3, which used to be stage 3 and 4 of the old classification, you now get very large, slow waves called delta waves. And this is what we call deep sleep. So you can see that as you go into deeper sleep, 2 things happen. The amplitude of the EEG increases and the frequency slows down. Also note that during REM sleep, you have a pattern that's like beta activation. You see beta waves. And this is why it's called paradoxical sleep, because the EEG looks like you're awake, even though you're asleep. What are the physiological changes taking place during this cycle? Well, during non-REM sleep, you find that your brain activity is decreased. Persons have good muscle tone, and the parasympathetic nervous system predominates with a decrease in heart rate, blood pressure, and increased GIT motility. The respiration is regular, and the body temperature is maintained. Let me talk about arousal, or what we call waking up. During non-REM sleep, you don't tend to wake up spontaneously. However, if somebody shakes you, that's an external stimulus, you tend to wake up. So let me just say that again. During non-REM sleep, you don't tend to wake up spontaneously. This is when you're in deep sleep. It's unlikely for you to wake up. But if there's a loud sound or somebody shakes you, you tend to wake up. Non-REM sleep has low association with dreaming. It doesn't mean that you can't dream at all, but the association tends to be low. And non-REM sleep is associated with some strange behaviors like sleepwalking and sleep talking. Well, in REM sleep, you see the opposite. You see you have increased brain activity with an EEG resembling that of an awake person, or even higher frequency. The body temperature tends to drift towards the ambient temperature. And what you find is there's a complete loss of tone throughout the body. There's atonia. And the only muscles that retain their tones are the muscles of respiration, the diaphragm, to ensure that you're breathing, and the eyeball muscles, hence the name REM, and the rapid eye movement of the eyes, and also the small muscles of the ossicles in the ear. The sympathetic nervous system now dominates with increased heart rate and blood pressure, and respiration becomes rapid and irregular, and there seems to be a lack of response of the respiratory system to CO2 changes. This stage of sleep has the high association with dreaming. It's believed most dreaming takes place here. And then you have penile erection and clitoral engorgement. Why this takes place, we're not too sure, but it does allow us to differentiate between physiological and psychological importance. If someone has psychological erectile difficulties here, they will still experience penile erection. In terms of arousal or waking up, here spontaneous is easy. And so most times when you wake up in the morning, it's because of the fact that you're in REM sleep. However, Awakening by external stimulus is a lot more difficult. This is where you might be shaking someone and trying to wake them up, and they somehow won't wake up. So let's just look at a little video summarizing all of these things.
Welcome to 2-Minute Neuroscience, where I explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the stages of sleep. Sleep stages are defined based on the measurement of electrical activity in the brain using an electroencephalogram, or EEG. An EEG represents fluctuations in brain electrical activity and voltage as a waveform of variable frequency and amplitude. The American Academy of Sleep Medicine classifies sleep as consisting of 4 stages. The first 2 stages involve light sleep. When someone begins to fall asleep, they enter stage 1, during which an EEG records low amplitude waves of mixed but mostly high frequencies. Next, the person enters stage 2 sleep. This is characterized by the presence of phenomena on an EEG known as sleep spindles and K-complexes. Sleep spindles are trains of high-frequency waves. The K-complex involves a biphasic wave that stands out from the rest of the EEG. Stage 3 sleep is also known as slow-wave sleep or deep sleep. In stage 3, delta waves, which are low-frequency, high-amplitude waves, make up at least 20% of brain activity. Stage 3 sleep is thought to be especially important to overall restfulness. Next, the sleeper passes rapidly back through stage 2 and stage 1 before entering rapid eye movement or REM sleep. In REM sleep, EEG activity resembles what's seen in stage 1 or restful waking. During REM sleep, the muscles are completely relaxed and limp, but the eyes are moving rapidly. This is the time of sleep when our most vivid dreams are likely to occur. After REM sleep, the person will sometimes awaken briefly, but then will move through the sleep stages again in order. Most people will repeat this cycle 4 to 5 times a night, with each cycle lasting about 90 to 110 minutes.
So that just summarizes the stages of sleep. Well, let's talk a little bit about what we know about the neural mechanisms of sleep. Remember, we started off talking about the reticular activating system and these long projection neurons. And you can see that these neurons are found within our reticular formation, the brainstem. And they are important for keeping us awake and alert. And you can see some of the neurotransmitters involved: acetylcholine, noradrenaline, serotonin, histamine. Incidentally, that's why when you take cold medication with antihistamines, some of them will make you drowsy because they block this histaminergic awake state. Now, there's a region within the hypothalamus called the ventrolateral preoptic area, VLPA. And we now know that that region promotes sleep. And what we describe to explain the mechanisms of sleep is what's called a flip-flop mechanism. These 2 regions seem to inhibit one another. So when you're awake, not only do the projection neurons keep you awake and aroused, but they also inhibit the ventrolateral preoptic area. But at some point during the cycle, the daily cycle, your circadian rhythm, the ventrolateral preoptic area turns on. And when it turns on, it turns off the projection neurons that keep you awake and then begins to promote sleep. So what you find is when one is on, it turns off the other. And that's what we mean by this flip-flop mechanism. So what then might cause the VLPA to turn on? Well, it seems that some of the neurons in the basal forebrain inhibit the VLPA. But adenosine has now been shown to actually inhibit those neurons that inhibit the VLPA. Now, adenosine is a byproduct of actively metabolic neurons. When neurons are actively metabolizing and producing energy, adenosine accumulates. So the theory goes that the accumulation of adenosine throughout the day eventually leads to enough adenosine to inhibit these neurons in the basal forebrain that inhibit the VLPA. Once this inhibition of the VLPA is removed, it now becomes activated and switches off the arousing signals coming from the reticular activating system. Interestingly, based on this knowledge, I now want you to look at the role of caffeine in sleep. Now, there are other centers that are involved, and these have come from studies. One of the studies occurred in dogs which had narcolepsy. Now, narcolepsy is a strange condition in which persons, and in this case the dogs, suddenly fall asleep in the middle of the day, and it occurs almost instantaneously. And in particular groups of dogs that were genetically suffering from narcolepsy, it was found that they lacked a particular neurotransmitter in the lateral hypothalamus. We now call this neurotransmitter the hypocretergic neurons, or orexins. Different names are used. And these also help to stimulate the brainstem activating systems. And it seems that this pathway is important for motivation to remain awake. So for example, you're feeling sleepy, but you're forcing yourselves to stay awake. This seems to involve these orexin or hypocretergic neurons. When these neurons are lacking, as I said, it does produce problems with sleep, one of which is narcolepsy. Okay, so we know a little bit about the mechanisms of sleep. Let's just now talk briefly about circadian rhythms. Now, circadian is derived from the Latin word circa, which means about a day. And it refers to the fact that humans, like many animals, have a biological clock. That means that throughout a 24-hour cycle, different things happen at different times. One of the most common things that we know about in medicine, aside from the sleep cycle, is the movement of cortisol, the pulsations of cortisol. We know that cortisol, which is important for preparing the body for activity, it's called a stress hormone, tends to peak early in the morning, and then it tends to decrease throughout the day. And that would be another example of a circadian rhythm. There are other rhythms in biology, like the menstrual cycle, which is a rhythm over 28 to 35 days. And then even things like hibernation, which is a rhythm over a year when birds migrate. Now, with regards to the circadian rhythms and sleep, we know that if humans are placed in a totally dark environment, that their cycle doesn't actually correspond to 24 hours. It actually gets a little longer, about 24.5 hours. And what this tells us is that there's intrinsic genetic mechanisms that are involved in the circadian rhythm, but these can be influenced by light. Let's just talk a little bit, or just use this video to tell us a little bit more about circadian rhythms.
Why do most people get sleepy in the afternoon? Why do they feel alert and energized around 10 in the morning? Well, there is something that guides how we feel throughout the day called the circadian rhythm. This is a rhythm that is naturally occurring and is built into the systems of the human body along with plants and animals. The term circadian rhythm was developed in the 1950s by a man named Franz Halberg, who derived the term from the Latin words circa, which means about, and diem, which means day. Therefore, the word literally translates into about a day rhythm. Even though the term is relatively modern, scientists have been aware of this rhythm for thousands of years. Both the Egyptians and the Greeks had some understanding of the circadian rhythm. There are several criteria that a biological circadian rhythm must follow. First, it must last around 24 hours. And second, the rhythm can be reset by exposure to light and heat. This phenomenon can be seen as we travel across time zones and get jet lag as our bodies slowly adapt to the new local time. Recently, it has been concluded that disruptions to our circadian rhythm can cause health problems. Chronic disruptions of the circadian rhythm has been shown to cause fatigue, insomnia, and is at times linked to obesity and diabetes. At this point, you might be wondering, What happens during this 24-hour cycle? What exactly happens during this rhythm? If you take a normal day and break it down from sunrise to sunrise, it may look something like this. In the morning time, our body experiences its sharpest rise in blood pressure and a reduction in the secretion of melatonin. These occurrences cause our bodies to feel awake. It has been recently discovered that enjoying a cup of coffee first thing might be counterproductive to your day. In fact, early in the morning, our bodies have the highest level of a hormone called cortisol, which makes us naturally feel more awake. The caffeine in coffee actually interferes with cortisol production. Your body makes less of it accordingly, and the body then relies more on the caffeine to feel awake. By this logic then, it is better to have a cup of joe in the early afternoon so that the body does not become as dependent on caffeine to feel alert. In the afternoon, our bodies have the best coordination, reaction time, cardiovascular efficiency, and muscular strength. This gives us an explanation as to why so many world records are broken in the late afternoon. In the evening time, our body temperature begins to increase along with the secretion of melatonin, signaling to our bodies that it's nighttime and it's time to go to sleep. We hit our deepest stage of sleep around 2 o'clock in the morning. Using this information is key to living a productive and happy life. After all, it's part of our nature, and the fight against nature is not easily won. Anyways, it's about 5 in the afternoon right now, so I'm going to go ahead and get ready for my afternoon run. Thank you for listening, and please click the button to subscribe.
So what controls these circadian rhythms? Well, we know the key structure appears to be a very small region of the brain called the suprachiasmatic nucleus. Now, it's part of the hypothalamus and is located just above the optic chiasm, the place where the two optic nerves cross, hence the name the suprachiasmatic nucleus. And there's a small branch, a small nerve that runs from the optic nerve to the suprachiasmatic nucleus, known as the retinohypothalamic nerve or path. And therefore, you can see there's a link between light coming through the optic nerves and the suprachiasmatic nucleus, which seems to be important in resetting the sleep-wake cycle and circadian rhythms. Here's a little video just talking about the work of the 2017 Nobel Prize scientists who got their Nobel Prize for their work on understanding what happens in the suprachiasmatic nucleus.
The 2017 Nobel Prize in Physiology or Medicine was awarded to Dr. Jeffrey Hall, Dr. Michael Young, and Dr. Michael Rosbash for their discovery of the molecular mechanisms controlling circadian rhythms. Because I'm not on the Nobel Prize selection committee, I can't tell you why they picked this discovery over a number of other deserving discoveries. But I can tell you what these researchers did to win their prize and why it is definitely worth the Nobel. Every morning you wake up and go throughout your day. You eat, you work, you get sleepy, and you go to bed only to wake up the next day and do it all over again. And you aren't alone in this behavior. Every animal and plant you've ever seen does the exact same thing. Every living creature on this planet is on a 24-hour cycle of activity and rest called the circadian rhythm. What's amazing is that we do this even in complete darkness. So how do we know when it's day and when it's night without the sun? Our 3 Nobel Prize winners had the same question, and to answer it, they used a very simple and unassuming insect, the fruit fly. These scientists worked on a gene called period that influenced the circadian rhythm of the fruit fly. One mutation shifted the cycle from the normal 24 hours to a shorter 19, and another one to a longer 29. A final mutation removed all rhythm from the fly, causing it to wake and rest at seemingly random times. But what was this gene doing? Was it making a protein that controls the timing? And if so, what was that protein doing? And where was this protein in the fly, or even in individual cells of the fly? And finally, how did it change the circadian rhythm? In 1988, a few of those questions were answered. A new paper came out that revealed that the period protein is in the nucleus of cells scattered throughout the body of the fly, and that period was found more during the night, with very little around during the day. This was the first hint that period was cycling in the same way that day and night do. From this protein cycling discovery, they came up with the idea that perhaps the period protein turned off its own gene. One of the easiest ways to turn on and off a gene is to have the protein that gene makes also be the switch master. The researchers thought that the period protein turned off its own production, which would lead to a cycle of period amounts. It's kind of like when your car shuts down because it overheated. Once it cools down, you can start it up again, but when the engine gets too hot, it'll shut off. Over the next 6 years, period was heavily studied to find evidence for this feedback loop. And in 1994, another player showed up that helped confirm the feedback loop was real. Another gene called timeless. The timeless protein worked with period to move into the nucleus of cells and stop more period from being made. Timeless is kind of like an employee with security clearance taking in an expert. Without timeless, period can't get into the nucleus. But period and timeless aren't enough to explain the whole cycle. The final piece of the puzzle came in 1998 with another gene called double time. The double time protein reduces the stability of period outside of the nucleus, which leads to a delay in the cycling. So the whole setup goes something like this. The period gene starts making the period protein. The double time protein limits the stability of period so it doesn't build up too quickly. Timeless shows up and starts teaming up with period to move into the nucleus, and period shuts down its own production. But once the amount of period in the cell gets too low, the whole process starts up again. A calm, cool, and collected 24-hour cycle. After the discovery of double time, the molecular basis of the circadian rhythm was well in hand. Scientists are still working out other rhythm details and how the circadian rhythm affects the rest of your body, but thanks to Dr. Hall, Dr. Rosbash, and Dr. Young, the molecular basis was no longer a mystery. But to really understand part of what made their work so special, you have to understand the time that they did it in. DNA was discovered in 1953, but it wasn't until 1966, 13 years later, that we really understood what all of those A's, G's, T's, and C's were actually for. They were coding for the individual amino acids found in proteins. The 70s brought the first animal gene ever cloned and the ability to actually sequence DNA. And it wasn't until 1983 that we found a way to be able to make more DNA really quickly using polymerase chain reaction, or PCR for short. Molecular biology was progressing, but it was still massively difficult to go from a physical behavior to finding out exactly which genes were responsible for it, let alone for a complicated behavior like circadian rhythms. The fact that the molecular basis of circadian rhythm was found in the 80s and 90s is incredible. Circadian rhythms control so much of our health that we can feel them get out of whack every time we hop on a plane and change time zones. The work these Nobel laureates did is beautiful. It's a great feedback loop that perfectly mimics our days and nights here on Earth. And the fact that they found the molecular basis for this at a time where molecular biology and genetic techniques were only just getting their start only makes the work that much more impressive. So congratulations to the 2017 Nobel Prize in Physiology or Medicine winners. And I only have one final question. Can you cure my jet lag yet? If you like this video, hit subscribe because I put out new videos every other Friday. And a special congratulations to Dr. Michael Young, who won this prize at my home institution of Rockefeller University. It's always great to see someone so nice win such an honor. So keep up the great work, Mike, and I'll see you all next time.
So, nice video explaining how we can solve complex neurological problems by looking at the genes. You would have heard a lot about melatonin. And what we now know is that melatonin is produced by the pineal gland, and you realize it promotes sleep. How it does that, though, is still under a lot of investigation. And that's all I'll say for now. But we do believe that somehow melatonin is able to reset the biological clock by affecting the suprachiasmatic nucleus. Finally, I'll just conclude by mentioning that as modern life has gone on and sleep cycles have been affected, sleep disorders have become more and more prevalent. Now, there are two types of sleep disorders. There are dysomnias, where people have difficulty sleeping enough. But then there are also parasomnias, where people have abnormal behavior during sleep. This is like sleepwalking and sleep talking, or problems with dreams. This is actually less common. In terms of dysomnias, one that's getting a lot of attention is sleep apnea, in which breathing repeatedly stops and starts throughout sleep. We now know there are two types of sleep apnea: obstructive sleep apnea and central sleep apnea. And this constant stopping and starting of sleep can mean that we don't get restful sleep. And it's now been associated with many, many health conditions such as diabetes, weight gain, heart failure, and even accidents. Hence, a lot of effort being put into diagnosing and treating sleep apnea. And just finally, let me mention again narcolepsy. Narcolepsy is an uncommon disorder, but it's important in sleep because the discovery of narcolepsy and the genetics of narcolepsy have helped us understand something about the mechanism of sleep. During narcolepsy, there's persistent sleepiness throughout the day, and then you can actually suddenly fall asleep, which is called cataplexy. Sometimes they get hallucinations and can have frightening dreams. And as I mentioned, it's due to a dysregulation of the protein, the neurotransmitter orexin. So that brings us to the end of our talk on the reticular formation, sleep, and coma. The reticular formation is an area of the brain that continues to undergo exploration as we try to work out exactly what it's doing. But as you have seen, it's important, very important for many bodily functions, especially attention, arousal, and helping to regulate the sleep-wake cycle. As always, until next time, take care.